In this video we're going to be talking about the major types of neurotransmitters that are found in the human body. So we're going to be concentrating on 3 types. So we've got glutamate, glycine and GABA. We're going to be looking at glutamate first. So one of the important, really key feature of glutamate is that it's an excitatory neurotransmitter throughout the nervous system. So it excites most neurons within the central nervous system. So once glutamate binds to its receptor, it causes excitation of neurons. They are then more likely to fire action potentials. Okay, so we must remember that glutamate is an excitatory neurotransmitter. Okay, there's 2 types of classes for glutamate receptors. So we've got inotropic and metabotrophic. Examples of inotropic glutamate receptors are NMDA, which are permeable to calcium ions, AMPA, which are permeable to sodium ions, and we've also got kainate. Examples of metabotrophic glutamate receptors are muscarinic glutamate receptors, which have 8 subclasses, which we won't, in this video go into detail. So this slide shows a typical glutamate synapse. So for this slide I want you to concentrate on how glutamate is synthesized. So I want you to understand from this slide how glutamate is synthesized. So as you can see here, we've got glutamate in this synaptic cleft there. So we've got loads of glutamate there. Because glutamate is charged, that means that it doesn't cross the blood-brain barrier in order to function. So therefore it must be synthesized in neurons from local precursors. So local precursor for glutamate synthesis is glutamine, right? Glutamine is released by glial cells. So the way it works is that glutamate is taken up by glial cell, which in this case is an astrocyte. So it's taken up and it's converted into glutamine by glutamine synthetase, right? After this process, glutamine is taken up into the presynaptic terminal. As you can see here, glutamine is taken up by the presynaptic terminal there, and it's converted into glutamate by enzyme called glutaminase. Right, so after it's converted into glutamate, glutamate is then packaged into these small little vesicles, synaptic vesicles, by a transporter called V-glute, as you can see there. Okay, once the glutamate is released, it binds to glutamate receptors, and glutamate is removed from the synaptic cleft by a transporter called EAAT, right? Then glutamate is taken up by glial cell, as we mentioned before, and the whole process starts again.